Yes, I heard a question back there. I see a lot of folks that I have never met before, that I'd love to meet at some point today. And I apologize to our speakers today because I walked in the headquarters and said, "Who are you?" Because I was expecting a lot of folks to come today that I hadn't seen before. So I'm glad to see that see some faces in here that I've not seen before. So welcome to our first Tyson Environmental Conference. I am Kim Medley, the director of Tyson Research Center. Thanks for forcing me to do that, Susan. I'm so at home here now. Hopefully all of you are as well. We have a great lineup of speakers today that we're excited to present. We do have a break in the middle, a substantial break. So we were kind of hoping for walks there. I don't know if that's going to happen with the heat today. It's kind of gross. But this is a new format for us. And I think I've told some of you in the room about this, the history of what we've done here at Tyson in the past. And for at least 15 years, we've had an annual summer seminar series that takes place weekly, and they're traditional sort of hour-long presentations in a relatively warm room after a long day in the field. There's some falling asleep, right? So we decided to do things differently this year and have some shorter talks on a variety of topics, and the variety of topics here almost capture the diversity of things that are happening out in our community this year. So we're looking forward to seeing how this works. And to that end, Susan has put together, in fabulous Susan style, a survey. I know it's hard to believe. But this is a survey that we really want to hear from you on. So on how this went today, your thoughts on doing this going forward, etc. So we've got a QR code set up there, various places throughout the room. So please feel free to scan that and fill out the survey so that we can decide what to do next year. Okay. I'm going to hand it over now to Susan, who's going to do our land acknowledgement and get us going. So thanks everybody for being here.
Hold up, sorry. There we go, wait.
It's okay. Okay. Thank you all for being here. We really appreciate you all being here at Tyson. We think it's a special place. But we do need to take a moment and do a land acknowledgement. So Tyson Research Center occupies land taken from Indigenous people. Our commitment to social justice and stewardship of this land calls us to understand this history. These nations include Osage, Quapaw, Sioux, Kaskaskia, Kickapoo, and Missouri. And today there are over 400,000 Native Americans living in Missouri. We make this acknowledgement because Tyson is committed to becoming an anti-racist environmental field station and to making science more diverse, equitable, accessible, and inclusive. It is vital to acknowledge the ongoing history of colonization and white supremacy upon which this station is built to begin the work of healing, reconciliation, and justice. Acknowledging the Indigenous history of this land is just the starting point to giving witness to the lives of these people and their impact upon this region. We call for the entire Tyson community to work together as we learn and grow and hold ourselves accountable to this mission.
For introductions.
All right, hi everybody. I'm Anna Wassel, and I'm going to be introducing our first speaker, Alicia Fox. She's joining us today from the Chicago Botanic Garden. Yes. This is Alicia. Say hi. Alicia is a research scientist working on integrating plant traits, communities, and the soil ecosphere to inform restoration as part of the National Science Foundation Biological Integration Institute. When she started undergrad, she thought she wanted to be a veterinarian, but a plant physiology course taught by her undergrad advisor at Elmhurst University opened her eyes to the weird lives of plants, and she's been studying plants ever since. She went on to earn her master's and PhD at Northwestern University in the lab of Andrea Kramer, where she studied plant, root, and shoot traits in the American West. For her postdoc, she ventured down to Florida to try something totally new. As part of a USDA ARS fellowship at Oak Ridge Institute for Science and Education, she studied the seed microbiome using applied machine learning and genomics. Alicia is dedicated to creating a better world for both plants and people, especially the diverse people studying plants. So we are very excited to welcome her to our Tyson community. Let's give Alicia another warm welcome.
This is so exciting. I get to see the microphone.
It's up to you.
Very high call. I don't think I can well, thank you all so very much for inviting me. I'm very excited to be here and just being here and seeing the community. I feel inspired. What I see here is beautiful and certainly things that I want to see in my community where I work. And I'm going to take some of these ideas back if you don't mind. It's not copyrighted, right?
No.
So very excited. The place is beautiful. The research you all are doing is so impactful and amazing. So thank you so much for inviting me. And so I'm very excited to tell you about this research that I got going on. My name is Alicia Fox. I'm at the Chicago Botanic Garden. I'm a conservation scientist. And what I'll tell you about today is exploring, let me read it, plant population trait variation and restoration context. Let's hit it. So goodness, my research program is about improving restoration efforts to ultimately help remediate habitat degradation and climate change. And I do this basically I'm just here to like annoy everyone, but I like to interrogate restoration practices and identify underutilized methods and ideas to improve restoration practice. So this includes thinking about what we do in our restoration and conservation efforts, but also who is represented. So making sure that Black and brown bodies are in the room and the knowledge that we have is brought to the table. And so I'm going to tell you today about a few stories that'll highlight variation in plant root traits and their relationship with plant restoration. And I hope to impress upon you all that intraspecific variation affects the choice of plant material when you are restoring a habitat. So first, what is intraspecific variation and what does it mean towards plant ecology? So there are a few kinds of variation we can be thinking of between clones, individuals, and material lines, probably other ones that I did name. But what I think about often is between populations. And so the mechanisms that get studied most are plasticity and genetically based differences. Am I doing this right? It's hard. Yeah, do you agree? Okay. Is it okay? Okay, sorry. And so plasticity being the response of a genotype being able to have multiple different responses based on physical and biotic influences. And so that can range from, actually for genetically based differences, we're also thinking about adaptation, mutations, genetic drift, and other things. But they can come about through the environment and through biotic interactions or influences, so like insects, pollinators, herbivores, and then interaction with other plants. And so I think a lot about between populations. So the reason why this is important to think about in the restoration context is because species that occupy a wide range of environments can exhibit interspecific variation. So we've got different environments that they might face in one area, don't exist in another area, and so there might be variation in what those plants look like, what they can do, and what that might mean for when we're using them in restoration context. And so when I think about what they look like, I think about functional traits, and so those are physical characteristics and physical, including morphological and perhaps also physiological, that influence their fitness, that affect and resulting in impacts on survival, reproduction, and growth. And so a lot of what I'll tell you about today is my research in the Western United States and the Colorado Plateau. Some nice pictures of sagebrush. They love sagebrush in the West. And here's probably in Utah. I took this picture on the internet.
I'm having in my own discipline, so sorry about that. And so one of the critical issues in the West is invasive species, and so particularly invasive annual grasses, and so this is a picture of seagrass Bromus textorum, so it literally grows like this, within the space of maybe like a bathtub that can be 13,000 individuals. They grow, yes, they grow pretty wildly. They have these dense root systems that compete pretty well with invasive species. So this is a can of one of them. It's really fibrous. They germinate when it's really cold and there's still snow out, and so they can beat out other native species, resources. If you can imagine, this burns really well. And so they have changed the native fire cycle from every 50 to 100 years to every 3 to 4 years. And native plants can't deal with that. So this is the dominant plant in the West, unfortunately. And so it's a problem. It's a conservation issue. It is a restoration issue. We got to think about it. So because of the way it does all of this, I'm really interested in the root system. Generally, and a lot of research and observations have shown that it does a really good job of looking out at native species. And so, you got stuff like this, and then sometimes you just got like one native species that's persisting. And I was giving the example earlier that when folks restore habitat in the West, they're happy to give one species to do their thing. And so, man, it's hard. And so, to me, one way to improve the establishment of native species, is to evaluate the native species characteristics and whether they can establish in these competitive sites. And so, I wanted to know, using a native bunchgrass, Elemis eloides, whether there was first variation in their traits and whether those traits resulted in differences in its ability to feed that invasive grass, cheatgrass, Bromus textorum. And so, this previous work has shown that the interface between roots is a crucial zone for interactions. And so for me, I was really interested in, wait, I gotta bring my thing. I was really interested in what are called root tips. So they're the ends of primary roots and lateral roots, and that's the source of where most of the nitrogen and water gets taken up by the plant. And so I quantified this trait as well as other traits. We didn't see much resolution above ground. And so we stuck with. So we wanted to look at this for resource reasons, and we thought, and the literature has suggested that that's a really important trait for competitive ability. And so we quantified the number of root tips in 8 Utah and Colorado populations, and the cheatgrass population. And we found that, it's kind of low, I apologize for the people in the back. All right, so we found, so the E's are the Elemis eloides. So these are, we just be like a whole run of the mill, about 10 mean number of root tips. And then we got cheatgrass over here, and so it's kind of got more. And then we had this extraordinary Elemis population. And I was like, what's going on? So I took this population, and I took one of these running mill populations. I was like, how do y'all compete with cheatgrass? And so we grew them up together. This is a picture, it's a greenhouse experiment. We established the cheatgrass first, about 275 individuals. Then we introduced, which is less pressure than they would experience in the field. So we established them first, and then we introduced the Elemis eloides seedlings, and we evaluated the survivorship of them. And oh man, they no mistake, like they all died. But we did see that some differences here. So the population at the top with more root tips survived in competition with cheatgrass at greater proportions than the ones with fewer. And so that tells me that, well, first of all, populations are not equivalent in their traits and the functioning of those traits, and that populations with certain traits might be suitable for restoration given particular goals. And so it's important then to evaluate the different populations and how they behave in these restoration, and particularly in these restoration contexts. So this is a little unsatisfying, but I don't really know why that population did what it did in comparison to the other ones. We did see the same thing in the field where it performed better than the same populations in the field. Collaborators are doing this work. We got those seeds from collaborators, so we don't really know about the ecology, what was behind why those why that population did what it did. And so we were like, let's take a closer look and let's be more intentional about knowing where those plants came from and what happened. And so I want to turn our attention to thinking about invasive species influence in that way. And so invasive species, terrible. They critically threaten native plant communities and reduce plant diversity. However, some native species adapt to the presence of invasive species due to a strong competition among some other mechanisms. And so from a restoration context, these seed sources could be a valuable feed source for restoration efforts to improve survivorship and competition with these invasive species. And so I wanted to know whether native species trait vary due to invasive exposure, and we use for another grass, Sporobolus xeroides, and which have been exposed to an allelopathic invasive species called Russian knapweed. And so let me tell you a little bit about this project. So we got some seeds from a collaborator, and so they had a collaborator at the University of Wyoming, and so they identified a population. The population that were 20 meters apart in terms of subpopulations, one without Russian knapweed, one with Russian knapweed, and then the co-occurrence was recorded for about between 40 and 75 years based on varying records. They took whole maternal plants, transplanted from the field to the greenhouse, and they grew them in separate greenhouses so they could prevent crossing. And then they collected seeds in the first generation, took those seeds, put them in the second generation, grew them up with seeds, made some more seeds, and then we use all those 4 accessions in our common garden experiment at the Chicago Botanic Garden. And we're focusing on the results from the second generation of seed production. And so we took some above and below ground measurements. Yeah, and then one of the things that I thought was cool about Sporobolus xeroides is that some of the primary roots were very fibrous and some were not. We termed them non-fibrous primary roots and fibrous primary roots, which is a terrible name. We'll fix it in post. And so what we found was generally native plants that were exposed to invasive species were larger than the ones that were not. For all the traits that we had, we only saw significant differences in the root traits and not the above ground traits. What? And so I think that that's indicative of creating stronger or perhaps promoting stronger competitors in those native species following invasive exposure. And I think that that could be potentially from the mechanism that the invasive species puts pressure on them. So there is a allelopathic, mostly below ground. It also has allelopathic in leaves, but their interface is pretty tight there. And so I do think that these populations may be able to serve in invasive context to improve restoration outcomes. But I know what you're thinking. How generalizable is this? Okay, great question. So I was interested in that too. And so it's a reasonable question because different invasive species have different mechanisms for which they influence invasive or native species. And so the ones that are below ground, for example, include also nutrient cycling, reducing resource pools, and affecting the biota below ground. And so to address this question, I performed a meta-analysis on studies that looked at the influence of invasive species on root allocation at different resource levels. And this resulted in 9 studies on 9 invasive species and 17 native species. There are a lot more studies out there that have done this kind of thing, but they often don't collect root traits. And so but when you do syntheses, you literally have to deal with what people have done. And so this is what I was doing, but I think that the insight that below ground allocation can give is pretty crucial. And so what did I find? I found that there's no difference between the plants when they had invasive exposure, when they didn't have invasive exposure, when there was ambient resources. Whoa, well, that's weird. However, when there was resource stress, the plants that were exposed to invasive species had far greater allocation to their roots than the plants that were not introduced to invasive species. And so that tells me that the root allocation differences exist, but they were only made apparent when there were stresses below ground. And I think that this is evidence that the native plants were responding to the invasive species. by allocating more below ground, and I think that this helps to provide more benefits to resource capture below ground. And so next, it's not just the differences in the trait means that matter, that are important, it's the variability in those traits. And so thinking about different seed sources for restoration, we like native plants for restoration. Actually, that's not, we do like native plants, but some people still use non-native plants. I'm on video, right? I like native plants, you use native plants. And so, but unfortunately, wild populations cannot meet the seed volume need. And so when I say that, I'm thinking about restoration in the Western United States, where millions of acres burn each year. And so we need millions of pounds of seeds to restore those sites, a lot of seeds. And so one way, a big ways that restoration practitioners have met this need is to use cultivars. So you grow cultivars up in an environment, you're able to do this at a volume that you can acquire lots of seeds and you can put them out into your restoration site. Oh man, but cultivars are, as we know, like they're developed through intentional selection, and by definition, this produces trait variability and genetic variation. And this is by design, right? And so a practitioner, if you're going to buy a lot of seeds, you want to know how, you want to have some sense about the phenotype of the plant that you're going to get. And that's what we want, right? However, by changing the trait variability, we don't know enough about that. The impacts above ground have been thought about, but we haven't thought enough about what's happening below ground. And so if the trait variation has changed below ground, that might influence how these plants behave in arid environments. And I would argue most environments. And so what I did was I wanted to ask whether variation in traits differ in wild collected compared to cultivars. And so I took I got another graph, pseudo-American wild populations and 3 cultivars. And I wanted to compare their morphology after growing them to the seedling stage. We can talk about seedlings as well, because I keep a lot of this works on seedlings. And so goodness, we took some above ground and below ground traits, and so I'm going to point to 2 below ground traits. But before I do that, I just want to say that the wild collections had greater survivorship than the cultivars, and they had greater survivability, greater trait variability in 6 out of 8 of the traits that we measured. I pulled out root tip counts. I told you I liked it. And this is the cultivar. So cultivars that produce fewer root tips than the wild population, which that's not great in a place where you need water a lot. And so that's something to think about. But also something that was interesting was that the wild accessions, the cultivars had greater root allocation than the wild accessions. But I think that points to thinking more, going beyond size, and thinking about how these plants are being influenced, by these processes. And so to sum up, I think that this is evidence that the cultivation practice can reduce trait variation, and that could hinder long-term restoration goals. And implementing methods like introducing heterogeneity in the cultivation environment might help to mitigate some of these issues. Next, new system. So I want to tell you quickly about a project that I've started on called the New Roots for Restoration. It's a collaborative ecology group across institutional NSF biological integration grants. So our goal across several institutes, some of you might recognize local. So our goal is to understand how traits are linked above ground and below ground, and how the soil biotics of the living and the abiotic, the non-living characteristics are influenced by the traits. It's complicated. And so we want to know what patterns emerge in restoration relevant and agriculturally relevant species. And so now I'm thinking about tallgrass ecosystems. And so I'm preaching to the choir. They're endangered, and restoration can help to mitigate some of the damage that has been imposed to them by humans and colonization. And so in this context, I'm trying to think about how recent restoration practice of mixing multiple populations can influence your outcomes. And so evidence has shown that having multiple genotypes, multiple populations can lead to greater local ground productivity, greater abundance, and higher trophic level organisms. But we don't really know enough about the long-term implications of combining multiple populations. there's evidence that some populations just literally end up on top and the rest of them have gone. And so if that happens, then we waste time, money, other resources, when we do these things. And so now, these are my collaborators, sorry about that in the back. These are my collaborators, and so we are testing out this is a pre-weeding picture, please ignore that part. But this is the plants of interest, the Valley Purpurea, Scotiacarpus scoparum, and Silvium integrifoldum, that we're trying to understand how population and genotypic trait variation influence these mixtures. And we want to go beyond the number of populations and the number of genotypes. We want to know what their functional traits are, and see how they influence the way these plants are interacting. And so this, we hope that this can provide emergent patterns on combining different populations in a restoration context. And this is a new study, so I hope to share it more soon. And I'd like to now close with some acknowledgements to folks who have helped me and funding sources. They are so generous. And I'd like to thank you all, and I'm so happy to take it. Oh, yes, please.
So were the cultivars that you did, were those ones that were grown for use in like horticultural? Why were the cultivars produced?
Those cultivars were produced for restoration.
Oh, okay.
Yeah.
That's different
Yeah,
than
absolutely.
native bars that you put in your garden.
Absolutely, yeah. And so one of them I didn't point out was the result of crossing, I think, 23 populations. So you would think that it would have maintained some kind of variation there, but it didn't. It didn't. So yeah. Something to think about for us in the future as we develop cultivars. Yeah. Any other questions? Yes, please.
So how exactly are these cultivars made? Like, do you pretty much force natural selection, for lack of a better term, force natural selection, or they become powerful enough to be a amazing species? If that's the case, what is the danger that, for lack of a better term, that becomes a basic species per se? These are
So if we were to take some of those populations that formerly were exposed to invasive species and we wanted to produce those, if they sort of became weedy and unmanageable, what's the danger? That can be a danger. I think one of the important things that this kind of work needs to do is to have interactions, understand interactions. So not just like of the one species or the one population that you're studying, is how does it influence other species? What might be going on? How does it influence other populations? Perhaps what is it doing to other organisms in different trophic levels? That's a good question. And I think that's something that should be evaluated, definitely. Other questions?
Awesome. Thank you so much. What?
High floating meeting control. Yeah.
All right, hi everyone. My name is Kaylee Arnold. I'm a postdoc through the Living Earth Collaborative. And I am very excited to welcome Dr. Asia Murphy today. So Dr. Murphy completed her undergraduate degree at North Carolina State University, her master's degree at Virginia Tech, and in 2021 earned her PhD in ecology from Penn State. Asia is currently a UC Presidential Postdoctoral Fellow at UC Santa Cruz. Her research broadly focuses on where are species present and why, how many individuals are there in a population, and what strategies do species use to coexist together. Aside from her academic work, Asia is also a photographer, a writer, and science communicator. Among many other projects, Asia is a co-founder of Black Mammalogists Week and has helped to create the Black and Indigenous Scholars in Mammalogy Award in association with the American Society of Mammalogists. If you have any questions or any similar interests, please feel free to find Asia after dinner or during dinner.
I'm surprised she actually found all that. I just gave her like a list of. It doesn't expect a short one, so. I don't have practice for that, so hopefully this works out. You can try pinning it if you want to hold it. We can switch. This. You guys getting it? Right. So just like what the title says, I'm going to be talking about resource partitioning across the world in carnivora. I didn't realize that we were supposed to be like talking about like our general being research. I usually use camera traps, game cameras to look at carnivore distributions. Also the distributions of other species like small mammals, birds. I have camera trapped in Pennsylvania, Madagascar, and Virginia. So done a lot of work across the world. But this is just like a meta-analysis talk. So basic question, how do species coexist? This is a question that a lot of ecologists like to ask and try and answer. Stainer in 1974 wrote this paper called Resource Partitioning in Ecological Communities. And he basically broke down, he did an analysis himself. And he basically broke down different ways different species will partition their resources to coexist. And when we're talking about resources, we're not just talking about food, we're also talking about space and time. So I'm going to do a quick example. Now, I started this when I was in my master's. I included xenomorphs in my thesis talk, and since then I've somehow included it in every single talk, including the job talk I did. I did not get the job. Yeah, so if you guys aren't familiar, Alien, it's a sci-fi horror franchise. First movie was in 1979, so we'll have the first species be the xenomorph, the OG. And then the second one is the neomorph from Alien Covenant, which was the last film in the franchise. Sadly, it doesn't seem like they're going to make a final in that one, so sad. But basically, we have the xenomorph and the neomorph, both species that are rather closely related, about the same height. How do they coexist? How do they use this space represented by this green box? Well, what they can do is they use different habitats. The xenomorph uses the screen spot in the box, and the neomorph only uses this blue spot, and they never cross paths with each other. Maybe they're active at different times. So maybe the neomorph is active and doing this thing at night when the xenomorph is asleep. Actually, according to canon, both species are pretty much nocturnal. So, but I just needed this example. And maybe they eat different things. So maybe xenomorphs only eat adults. And again, there was no babies. That's just an example. But if we're looking at real species, we have the margay on the left and the ocelot on the right. Two species that are pretty closely related, South American, Central American cats. And we have margins, which are largely arboreal, using the canopy and the mid-story, whereas the ocelot is purely terrestrial. So they'll do their hunting in different levels of the vegetation structure. We have brown-tailed montseras and broad-striped montseras. These are two small carnivores that are found in Madagascar. And the brown-tailed montera is only active during the day, whereas the broad-striped montera is only active at night. And then we have different diets. So Fusa, which is the largest carnivore in Madagascar, the largest native carnivore, and the Falonique, which is on the right. And lemurs, Fusa are pretty well known for eating lemurs, but also eat small mammals, birds, etc. Whereas the Falonique will actually eat only insects and earthworms. That's why its muzzle kind of looks pointed like that. And looking at these different ways of coexisting and partitioning their resources is important because humans are taking up a huge amount of the kind of spatial, temporal arena that species can use. So we've got humans either taking up space through urbanization or just by going outside and being active. Studies have shown that different species will be sensitive to human recreation and activity and actually move away from trails and things like that. And then you also have them taking up this temporal niche in the daytime, even going up into the midnight, depending on the light and where the location is. But if you have humans taking up this huge amount of space and time, pie or pizza, whatever it is, deep dish, you leave this little bit for all the other species to use. And this is what we call coexistence. These studies have seen that different species will actually become more similar in their diet, their spatial habitats, their temporal activities, the more urbanized the surrounding area is. So, like I mentioned, I just did a meta-analysis. We're looking at a whole bunch of different papers and trying to figure out what influences whether two species will partition space, time, and diet. Because I'm interested in carnivores, I focus largely on carnivora, but I also included opossums, Dibelomorphia, and Dasynomorphia, which are the Australian carnivores, Tasmanian devils, spotted voles, etc. I used Web of Science and I searched up 43 phrases, different variations of competition, interference competition, overlap, niche partition, etc. I ran through thousands of papers, and I basically narrowed them down to whether the results were actually statistically significant. So I needed to see some sort of test and a p-value or something like that to include it in my data. And I ended up with 339 final papers to look at these different relationships between the factors that I'm about to list and whether species were partitioning this resource. I use structural equation modeling. It's basically just looking at correlations between different variables. I did some analysis in R, the piecewise SEM. I coded the data as one for partitioning. So if there's like two species use, one species use like a riverbank and another species use the open plains, that's spatial partitioning, that includes as a one. 0 for normal partitioning, I used a steady number as a random effect. And the different covariates I looked at, there are a couple. So I wanted to look at whether species that were really different in body size would partition their resources. So you have tigers right here and a leopard cat on the right. And, wondering, since the tiger is so much bigger than the leopard cat, would it be eating the same items? Would it be using the same habitat, etc.? I also looked at the shared taxonomic family to sort of get into those little bits that I could include like genetics in. So we have tiger and leopard cat both felidae versus tiger and the sunbear, which is two totally different carnivore families. I looked at latitude, so different pairings depending on where the sites were. Thinking that maybe like the closer it is to the tropics, the more even the sunset and the sunrise hours are. Maybe there's no real changes in temporal partitioning. Maybe there's differences in depth depending on where the locations are, etc. Also look at species richness. So maybe species are more likely to partition their resources depending on how many other carnivores there are in the location. So leopard cat, clouded leopard, and that's the banded civet. I had to include the civet because I love them. And finally, I also included a categorical variable that looked at whether they were already partitioning something in a different dimension. So if I was looking at whether these two species spatially partition their resources, so using different habitats, I included whether they were active at different times or whether they used different resources as food. And for the results. So out of all the papers, spatial partitioning habitat differences took up most of the studies. The next one was diet, so a lot of SCAP, eDNA, people studies. Across the globe, United States is most common, which is definitely an artifact of my searches. You know, I'm looking only in the English language. I'm only using web science, so maybe there's differences in publication rates and which papers are actually archived versus which aren't. Out of the countries, United States again was the most common. Brazil, South Africa, Australia, and India have the following number of studies from there. In total, out of those 339 papers, I ended up with 1,800 species pairings. And again, most of those species pairings were looking at differences in space use or in habitat use. The next one in this case was temporal, so different hours of being active. And what I saw was that in those species pairings, there's a lot of species that were either cats in Felidae, dogs in Canidae, or weasels in Stellidae. And if you compared that, if you looked at these families and looked at the number of studies they were present in versus the number of species that there actually exist, dogs, Canidae, Felidae, Canidae, the Stellidae, the darkish blue ones are all overrepresented in my data set. So there's not a lot of studies on Madagascar's carnivores, which are declared, or on Priodontid, which is a lindsay. And I just learned about that yesterday. I did not get any papers on that, so I was like, oh, okay. Out of all the species, red foxes have the most studies, so they were present in 73 studies. And looking at the geographic range, that kind of makes sense, they're all over the place, so it's likely that they'll be picked up as a species to look at. The next one was coyotes in 58 studies. Leopards in 55 studies. Pumas in 38 studies. And then lions in 31 studies. So obviously, if you're looking at the ranges, lions shouldn't have that many studies based on where they're present, especially compared to other species that are a little bit more common or more geographically widespread. And to look at the actual structural equation modeling results, again, I'm looking at whether the two species in a pairing, margins and ocelots, partition diet, use different species or different ages or different sexes of individuals. And what I found was that one, the factors that I had in the model did not actually explain dietary partitioning well. So R-squared is basically a measure of how well the model is explaining the variation in the data, and it's only 0.4. So It's not a lot. But I did see that, species that are in the same family, so red fox versus the coyote were less likely to partition their diet. And then species that have a larger body size ratio, so tiger versus leopard cat, are more likely to partition their diet. If you're looking at partitioning space habitats, this is a little bit better. R-squared kind of looks at 0.61 of the variations, so I really like this one. What I saw was that actually if the species are partitioning their space and habitat use, they also had different diets. Species in the same family were more likely to use different habitats or different spaces. And this, I don't even want to explain this. And then finally, the time, again, it doesn't really, like the factors that I have, doesn't really explain temporal partitioning all that well, fortunately. But species that have different body size ratios, larger and smaller carnivores, are more likely to be active at different times. So basically just wrap up, again, species that eat different foods use different areas, that makes sense. If you're in an open plain, you're gonna be eating gazelle, and then if another species is in a forest, it's gonna be eating monkeys or something, you're gonna have different diets. Species that are in the same family are more likely to use different areas, which is pretty interesting. I assume that it's because, like, if you're in the same taxonomic family, you might have similar morphologies, so you're more likely to be eating kind of similar things. And so you might want to use different habitats, or maybe it's just easier for species, more like, I don't know what's called, conservative, more efficient for species to be using different habitats than to totally evolve a different way of eating. That's possibly it, I don't know. Again, my factors didn't explain dietary or temporal partitioning very well. This might also be a case of small sample sizes. So my spatial and habitat data set was like over 1,000 species pairings versus the diet and temporal partitioning ones, which were like 600 and 300. So it's not a lot of samples, unfortunately. And then an interesting thing was that 52% of the carnivore families are understudied. Again, a lot of people are looking at Canada, wolves, foxes, coyotes. A lot of people are looking at lions and tigers and leopards and stuff like that. So people should focus on civics. And that is basically it. Yeah, see, I told my talk, it's gonna be really short. I'd like to thank UC California's Commercial Postdoctoral Fellowship Program for funding me while I was doing this. And I guess my advisor, because I totally dropped this project and I didn't want to deal with it until he asked me about it like 2 weeks ago, and I was like, all right. So yeah, and that's it. That's all I have. Yep. Yeah.
Thank you. So great talk. I'm in a huge amount of work doing type of analysis. I'm wondering, from your meta-analysis, or maybe extrapolating beyond that, is there any evidence that like species that of greater conservation concerns have maybe like a less successful partitioning of resources with other carnivores in their area?
I have never even thought of that. It's actually interesting.
And I'm guessing conversely to that, like, I mean, there's a upward bias with species that are being part of these studies.
Yeah.
So I'm wondering if species of very low conservation concern are more likely to also be strongly producing resources than others.
Well, I mean, there's different ways to sort of look at it. I'm definitely interested in using that status as maybe looking at their overrepresentation and underrepresentation in the data set. A lot of species of least concern are thought to be, like, you know, expecting to be generalist, pretty common, using all sorts of things, et cetera, versus the more endangered species being more specialized. But it's possible that there's differences. So can I actually include that? next round, so thanks. Thanks to you.
So your research mostly, like you focus mostly just like using online resources for this project, right?
Yeah, I just downloaded papers.
Are you like, would you be like hoping or like wanting to like observe the actual like studies of conditioning in person?
Run that back again, what did you say?
Like, would you like to do the actual like conditioning studies like in person, like out in the field?
That would be fun. But it would require a lot of money. So, yeah. It's always good to get in the field, get results to it, but cool thing about meta-analysis is you can take what everybody else has already done and look at different things, get new patterns. You.
So kind of thinking about like the coexistent squeeze. Do you, I don't know, I might be asking you to speculate too much, but like of the three kind of methods of partitioning, you talked about like space, time, and diet, like which one do you think would be more resilient to coexistence under that anthropogenic factor?
Oh, God. Probably temporal partitioning, because temporal partitioning does also have to do with morphology, like especially, with strictly nocturnal species, their eyes are gonna be totally different from strictly diurnal species. So it might be that, but I was hoping I would find something. So, somebody else had a question.
Oh, I had a question. I've never heard of partitioning studies. I wondered if you could maybe tell me a little bit about the methods. Like, is it a combination of like maybe camera trapping and then diet, like poop, looking for poop, and I mean, like, how do you do these studies?
So there's a whole bunch of different methods that I saw in these studies. A lot of people did use cameras, especially when they were looking at spatial and temporal partitioning, because cameras, they'll give you the time that the picture is taken. And if you have a location of the camera, you have the space and the habitat as well. But they also looked at telemetry, GPS collars, etc. The diets, it was largely scat studies. And it was largely like people picking out crumbs and stuff, but more recent papers have things like eDNA to look at it. I hope that, okay.
Thanks.
Yeah.
In terms of like the species of same family that like use different areas, do you know if that's a factor like the availability of other factors, or is that just like
I don't know for sure. It could be.
Into your experience, I guess that's scientific. You made a guess. What would you say the best mating move is?
Yeah. Okay, so I really like Aliens because that's the first one I saw when I was like 4 or 5, and my dad was watching it, and I wasn't supposed to be watching it, but I was sitting up behind him and was watching it. But I like the Prometheus and Alien Covenant because the focus on David as a villain is really interesting. I wish you would finish it, but I don't really want to. So Aliens, and then after that, Prometheus. Yes.
So what are some methods you're thinking about in the future for accounting for over/underrepresentation in the studies for certain species?
Are you asking me personally? Okay, so I don't think that I can really do anything about papers that are already published. I kind of mentioned it because I was hoping that, if I did publish this, I don't know if I want to write it up or not, that, people will be like, oh, these families are little, like, there's not a lot of research on these families, so maybe I should go out there and look at this. This is a master's project or a PhD project or whatever. Yeah, I mean, I could look at different search engines, so like Google Scholar. I thought about JSTOR, but like its abstract download thingy is just like so a support. Like I hate it. You have to like click on each individual paper and download abstract individually, whereas on science, we just download a whole bunch of them at one point or one time, so. I don't know, I'm not sure. We'll figure it out.
So I have a follow-up sort of thought to the methodology. So generally, to, I guess, filter on the p-value, I'm wondering if that's the space.
Yeah.
Because there could be studies that, I guess this is called like the Faujo problem meta-analysis, that people don't publish studies, but it is classified.
Yeah.
Yeah, and then so filtering off of those, is there they might be represented in those studies that will miss, but also
Yeah.
filtered on.
Yeah. So, I thought of doing descriptive, I actually included a lot of descriptive stuff in the earlier version of this, but. I wasn't sure how to go about it,
Yeah.
so I just decided to use this whole stuff.
Right.
Might, but I have to go back and do the descriptive stuff, but yeah, you're right, a lot of. Papers that don't show anything are hard to publish, so it's likely that this is overestimating patterns and, amounts of partitioning that we see.
Thank you. No, I have no idea at the moment.
Good, batteries also, I might want to start doing a break.
I can change it right now.
I mean, it has a lot, I can find. Okay, not working. Oh, muted. Do I hit that? Okay, okay. Hi, everyone. I am here to introduce Dr. Frank Tyson. He is an associate professor at University of Missouri, Kansas City. And he actually has a whole slide introducing himself, so I'm just gonna pass it off to him.
All right,
It rotates through there.
thank you. All right, good afternoon, everyone. First of all, I'd like to thank Kim again for inviting me to be here. It's really a pleasure. And also I want to thank Katie and also Susan to be the two this morning. What a fantastic facility. You guys are so lucky. Yeah. All right, so I'm a family scientist. I'm gonna have one slide to introduce myself next slide, okay? And here I want to just, I changed the title a little bit because I got some information from Kim that we have really very diversified background of students and also postdoc researchers. So today when talking about the climate change from global to local, I'd like to introduce you some kind of tools that scientists, climate scientists utilize to do climate change predictions and projections. Right, we're going to go to two scales. One is from global perspective. The other one goes to the local or the regional scale, right? And I really have a pretty hard name to pronounce. My first name is Denton. Some people might be not able to pronounce it very well, but I do have a very simple and cool last name. I shouldn't say cool name. It's a hot name. So I'm not sure if you guys like the hot weather, sunny weather today. If you like it, I take the credits. If you don't like it, I take the blame partially. All right, yeah, there you go. This is my first slide to introduce myself, and I put some kind of animals over there, and just give you some idea of my dark science academic journey. And first of all, I got my PhD in climate science from Department of Earth and Environmental Sciences from UC Irvine, and that's an teacher. All right, and then I kind of transformed to a bear, and I went to UCLA first as a postdoc researcher, and
assistant researcher over there. And then a couple of years ago, about almost 7 years ago, I moved to Missouri. And as you can tell, I transferred from the bear to a kangaroo for some reason. And I was hired as assistant professor. Right now I'm associate professor over there. As I mentioned, I'm a climate scientist. I do lots of work on climate studies. All right, this is my introduction. Well, yeah, I just first of all give you an overview about what we're going to talk about today. As you can tell, 2 parts. Okay, first part, we're going to talk about climate change from a global perspective. I'm going to introduce some kind of tools we utilize. And then the second part is that we really care about climate change from a global perspective. But for each individual of us, what we care the most is not really something happening in, for example, Africa. For example, over Antarctica, right? We really care about what's going on in our cities, in our neighborhood, right? And then for the second part, we can go down to the regional and the local scale. We can introduce you some other tools that we utilize to study some kind of regional or local phenomena, such as the urban heat island effect, which is something that I have been working on during the past few years. Of course, we are focusing on the Kansas City metropolitan area, but good news is that we just got some funding. We're working with Rachel and Kim. We're going to look at something happening in the Great St. Louis area. Hopefully, a few years later, I'll come back. I can present some results on that study. All right, so let's start with the first part. Okay, let's look at the global climate change. And first of all, I don't. Okay. Thank you. All right, and here let me start with this one. So this is kind of a report released by NASA at the very beginning of the year. But at the very beginning of the year, you can release the kind of report to tell you what's going on, what happened during the past year. All right, you're going to rack all the years during the past 140 years or so, because that's the time we have the network observations. All right, so what I show you in this picture, in this animation, that for each about 5 years, even to the average for the global temperature, surface temperature. Right, what we see in this, sorry, let me do it again, one more time. Where's the mouse?
It's on the screen. On the big screen.
Thank you. Thank you again. All right, what we show you here, this is kind of the animation tells you the evolution of global temperature. As you can see, this is kind of at the very beginning of the observational period. You see lots of blues, right? This tells you we call the negative anomalies. What do you mean by anomalies? When you look at each year's temperature, and then you calculate the long-term average, then you subtract that long-term average. Well, that gives you sometimes you got positive values, which tell you, okay, that year or those years particularly is going to be warmer than average. If you're going to see this kind of the blue colors, which means it's a negative value, it tells you, okay, for that particular year, particular years, it's cooler than average. What we see from this global map, we see that usually at the very beginning of the observation period, we have lots of blue areas, right? Which means those years tend to be cooler than average. Well, if you go closer to the most recent decades, we see lots of warm color, which means, okay, we have lots of warm temperature, right? And then according to, all right, here, according to the NASA report, it says here, when we look at last 10 years actually were the warmest decades on the record, and particularly last 5 years, actually, they are collectively the warmest year in the modern record, right? And here you see here for this picture, for example, here, let me just jump to this one. This is just a snapshot of what's going on for last year. Look at here, this picture, we see lots of positive values, right? Which means it's kind of warmer than average, right? But we definitely see other, some areas, instead of one red colors, like for example, over here, Pacific Ocean, you see some kind of cool colors, right? This tells you, okay, maybe, kind of cooler than average. That's corresponding to the so-called La Nina event. However, this year, there's a pretty strong El Nino event, which means this area is becoming more red this year. But anyway, this tells you what's going on for the global coverage, right? And then what we can do a global average. We take each of them land or ocean or each way cell equally, and then we do an average. Right here, I'm going to present you, this tells you a time series of the globally average surface temperature during the past 140 plus years. What do we see over here again? This one tells you negative values, cooler than average, right? Look at here, what's going on over recent couple of years. What do we see over here? You see lots of red, deep red colors, right? It tells you, if you look at here, global as a whole, we know this is happening, right? And here we can basically get the conclusion that, okay, the Earth's average temperature has been risen almost about 2 degrees Fahrenheit compared to the very beginning of the observational period, right? That's the first message. The second message, as you can tell, from time series evolution, you can see the warming trend is especially evident in the past few decades, especially after World War II, right? And here, as I showed you, this kind of curve, this straight line just tells you this kind of a steady warming pattern, right? So what do we call this? We call it global warming, right? The Earth is on fire, right? So, however, when we talk about the climate change nowadays, warming is only one piece of the signal, right? Because of warming, what are we going to see? We see lots of fires, right? Especially in the past few years. And here, look at this picture. This picture, it looks old, however, it's not really old. It's about 3 weeks ago. Right? This picture from New York City about 3 weeks ago, the Canadian fire, and then New York City has become one of the polluted cities around across the whole world, right? So this is a consequence of warming, because you have more of this and wildfires, and then you're going to have a big impact on people living in the cities and also in the rural areas, right? And also because of warming, look at this guy over here. He has been stranded in over a small piece of the ice. This has been utilized by many scientists as a kind of an iconic picture to tell people what's going on about climate change, right? And here the idea is that because of the warming, those ice snow over the Arctic and Antarctic area is starting melting, right? And here part of it is they can swim, but unfortunately they are not really good swimmers, right? They have to grab a piece of ice to stay, and then unfortunately now they in the recent past few decades of years, actually we've seen more and more drawn polar bear around the region. So this is the kind of consequence of global warming. And also because of global warming, you see the large scale circulation pattern change, while some areas tend to receive lots of rainfall, actually now they become very, very dry, right? This is kind of severe drought events. And as we know, the drought definitely have a huge impact on the economy, especially agriculture, right? As this slogan goes, food grows where water flows. No water, no food, right? And then also here, and also, so, however, in some other regions, during some, even for the same region, but different times of years or different time of years, they're going to see, look at here, what a huge contrast between that picture and this one. Actually, this is not that old. It's about 4 years ago. If you recall, back in 2019, you know, here, this is showing you the picture of the almost reached the dam over the Missouri River. Okay? And here I'd like to just give you the idea about how this flooding has been impacting the area. This is just right before and after the flooding event. As you can see here, this area is supposed to be green, which means lots of crops over there. Well, it's just one after 1 day period of time. Everything is under the water, right? So this is kind of another, when we talk about climate change, we are not only care about what's going on between this, let's say, the recent decades compared to 100 years ago. This is so-called long-term climate change. However, in addition to long-term average change, there's also extreme change. This is a perfect example that tells you those kind of extreme weather events, such as flooding, such as droughts. And also talk about those kind of extreme events. We have to think about what's going on in the summertime over Atlantic Ocean basin, right? If you can tell, this Florida is over here. And here we have 3 hurricanes. But this is not really that rare. Actually, it almost happens every year. But in the recent past few decades, scientists just realized, okay, we have more and more this kind of frequent, you know, very strong, you know, hurricanes, especially the landfalling hurricanes. Then it has a huge impact on the people and also civil infrastructures. So based on this, I can tell you here, we are talking about global warming. However, we need to get a more general concept. It's not only warming at all, right? It's all about climate change, right? And here, just try to summarize what we just talked about. So here we have lots of warming over the land surface, in the atmosphere, and also in the ocean. As a consequence, you can just look at it here, those ice and the snows over the high mountains. over the Antarctic and Greenland, they have been loss of those kind of huge pieces of snow and the ices. Okay, and then what's going on with the ocean? Actually, this morning we just talked about the Florida coast, the temperature over there. We are not talking about air temperature. We are talking about the ocean temperature. It's almost 90 degrees, right? It's become a hot tub, as Mia just mentioned, right? So as you can tell, okay, there's lots of things going on in the atmosphere of the ocean, and here it has been a alarming message to people. Wow, this is not really kind of only things. It's real. Right, but here, come back to my point, and here, still, I think, global warming is still the ultimate driver of all trying to change signals, right? And then, because of warming, you're gonna have lots of consequences, right? Unfortunately, those are usually are negative consequences to human beings, right? And then come back to the picture that we just saw, and here, I think I'd like to, just like I say, I do not have enough time to talk about the mechanism behind why you have a global warming signal, but here I think it's kind of scientifically grounded, and most scientists, the majority of scientists believe that those warming in the recent decades especially is because of the human activities, especially because we, through the burning of fossil fuels, we release lots of carbon dioxide and the other greenhouse gases into the atmosphere. Those greenhouse gases, they are accumulated on the surface ground, they're going to trap lots of heat. Instead, when they're going outside, they're going to trap all the heat. That's the reason why around the surface, you're going to see lots of warming. That's kind of the reason behind it. And also to verify that kind of conclusion, actually, I'd like to introduce another kind of iconic picture. We call the heating curve. And here, this is Dr. Charles Keating, used to be a professor from UC San Diego. More than 60 years ago, back in 1958, he set up some kind of instruments over Hawaii. The Mauna Loa observation in Hawaii, they started reporting the carbon dioxide concentration in the atmosphere. This is time when he pioneering started back 60 plus years ago. As you can tell, concentration over that time period is about 300 ppm part per minute. Nowadays, we have here steady increase is already past 420 parts per minute. If you're going to be able to draw a line over here, for example, with this picture, you can see, all right. Yeah, the increase of carbon dioxide is responsible, at least mainly responsible for the increase of the global average temperature, right? So this is has been scientifically grounded, okay? And here we know warming is happening, right? This is the data, this is true, ground truth observational data set, right? Warming is happening, and warming is continuing, and most importantly, warming has been accelerated, right? So this is based on everything on our observations, okay? And here, we know the climate change is happening at an unprecedented pace. It is a challenge not for our generation, my generation, but for your generation and also upcoming generations as well, right? Luckily, we do have lots of more than 100 years of data set tell us what's going on. But then you might think about another question here, how about the future? Okay, can we predict future climate change? The answer, of course, is yes, right? But here is something I'm going to introduce you. Actually, yeah, first of all, I'm going to introduce you to the so-called IPCC, Intergovernmental Panel on Climate Change. This is an organization by WMO, the World Meteorological Organization, and the UNEP, United Nations Environment Program. They set up almost 30 years ago. They set up the IPCC report. The idea is that as I come. All right, good enough. I think basically every about 5 or 6 years or so, they release a climate change report, tells you, hey, basically what has happened in the past, what might be happening later on. And here, this is the newest one, right after COVID, they released a number 4 assessment. In that report, twofold. One is, okay, we're going to try to summarize what's going on during the past, let's say, 5 or 10 years or so in the climate change research community. And most importantly, we want to do some prediction for the future. Utilize some tools that we introduced over here. And here, this is called the global climate models. And usually for us, we call it general circulation model, but we also kind of abbreviated as GCM. We use GCM to do the climate projection. So what do you mean by the GCM? First of all, those global climate models, they are computer models which solve a bunch of mathematical equations representing the scientists' best understanding of how climate change, how climate system works. Okay, for example, here, this is one of the complex equation. You are not necessarily remember what this equation is about. All right, this actually tells you this is one of the equations that tells you here scientists try to utilize the equations all right? Based on the physical law, try to understand how the air moves from here to there, how the heat transfer from here to there, all right? And then we are trying to solve those equations by some initial condition. Then those GCM will spit out some kind of snapshot, tells you what's going on for the current temperature of the other variables. For example, here, what I show you over here is a kind of just give you the three-dimensional view about the atmosphere. Right, atmosphere, we have longitude, latitude, we also have a height, right? That's gonna be kind of have a bunch of cubes in the atmosphere. For each cube, they have a uniform information about the temperature, wind, the humidity, and pressure. So what we show you in this picture, for example, it's pretty clear that we definitely see some kind of relatively warm temperature over here, right? Some relatively cool temperature over here. And this temperature there is relatively cool, and also those arrows tells you what's going on for the surface wind. Okay. And we can use this GCM to simulate past climate, and most importantly, we are able to project the future climate change. So then how are you going to do the projection? It's kind of complicated, but here essentially we're going to try to prescribe some of the carbon dioxide and those greenhouse gases in the model, and then we can tune the value of the concentration of those gases. For example, we can increase from about 400 ppm to 800 or 1000 ppm, and then we see how the climate model will respond to the increase of those greenhouse gases, and then they help us tell, hey, what's the future climate might be looking like. All right, and here I skip this one quickly. Well, come to this one, and here, look at here, where we are now, we're going somewhere around over here, right? And here, when we look at the end of century, right, we have a different kind of curve. Tells you different, we call them different greenhouse gas emission scenarios. Okay, to better understand why I'd like to introduce this one. This tells you the concentration of carbon dioxide in the atmosphere. Look at here, compared to the nowadays, there's one possible scenario. You're going to have almost triple the concentration nowadays. Well, we have another scenario, which is almost nowadays. Well, this so-called one of the mitigation scenario, the other one is just so-called the physics usual scenario. You may expect when we have two different scenarios, we might have different temperature distributions at the end of the century. Well, this one comes from the IPCC report. When we look at it here, right, it tells you the business that the usual scenario at the end of the century. Look at here, the temperature is going to be more or less 4 degrees warmer than nowadays. But if we have this kind of mitigation scenario like this blue curve, it tells you, okay, temperature still increase. That's simply because even though you stop greenhouse gas emissions, those gases put into the atmosphere during the past 200 years or so, still living in the atmosphere. They're still going to try to further warm up the atmosphere. So still we have 1 degree temperature increase. But, and here this picture just gives you the idea about the two scenarios. To the left, that's the mitigation. To the right, is going to be the business as usual. As you can tell, they are essentially very, very different, right? And here just try to highlight with that substantial mitigation and without. The choices we made will create very, very essential differences. All right. So I try to summarize a little bit. And here, the global climate model are very useful tools for us to invest in the past and present climate change, and also they have been utilized to project the future climate change. And also we know that human-induced greenhouse gases will mainly respond for the warming in recent decades. They are going to continue to play a very important role in shaping the future climate. Right? And the anthropogenic, this is one of the most important so-called human-induced forcing. And not the forcing is going to be due to the human activities, the so-called land use and land power change, right? And that's usually happening on a relatively smaller scale, not really kind of global scale, right? Or more in the local and the regional scales. We're going to examine this from a regional local perspective. That will also lead us to the part number 2. I realize I don't have enough time, 5 minutes? Oh.
8.5 minutes for no questions.
Okay, thank you. All right, and here when we talk about the global climate model, they are really useful. However, because they are global coverage, the resolution is going to be really, really low. All right, and here, for example, here I want to show you the typical resolution of global climate model is about 2 degrees, roughly about 200 kilometer by 200 kilometer resolution. And here I want to utilize it here the continent of the US as a good example to tell you what the resolution is for the typical global climate model. Here, first of all, I apply some kind of green. You're able to see the green cell, right? So this each of the green Green real cell is about 200 kilometer by 200 kilometer. And if we just focus here in Missouri, as you can tell, we can probably only use about 4 to 5 gray cells to represent Missouri, right? Which means for each gray cell, they have one single value. However, we definitely know it's not true, right? The resolution basically is the main issue. The idea is that just highlight a little bit, and here to the left, that is what we can get from global climate models. Well, usually for this kind of right-hand side, that's basically the information we want to get. Usually here, we want to down to our neighborhood, right? We want to look at here probably a few miles, a few kilometer resolution. The idea is that there's a huge gap between the left and the right? To explain the resolution issue, I want to give you the idea. Who can tell me what's going on from that picture? Look at here, this picture. That's a bit of an orchid flower, right? You can clearly see the difference. This is kind of resolution issue. And hopefully that will help understand this is the information we are able to get from global climate model. Well, this is something we want to achieve. There's a huge gap, and then to bridge the gap. This is something we call the climate downscaling studies. Okay, this is really technical part. I'm going to skip right through this one. The idea is that this kind of very useful procedure by climate scientists trying to regionalize those climate change signal. Not they are getting information from a global model, global resolution, but eventually you want to look at your neighborhood, all the regional and local scales. Okay, and here actually this has been the technique that we utilize a lot to stop many problems. One of the problems is the urban heat island effect. Keeping the same whole urban heat island, basically, I try to use not this type of 4 words. Okay, urban warmer than rural. Okay, this 4 word tells you what to mean by the urban heat island effect. Well, here quickly just give you some idea about what we have been doing in the past few years. We look at the Kansas City, the urbanization. This is the picture tells you what's going on for the Kansas City. In fact, about 50 years ago, the red tells you the urban area, while the rest of the non-urban area. Okay, here, this is about a few years later, and then look at it here, the further expansion of the urban areas, right? Definitely this kind of a local or regional phenomenon, right? And here, when we look at the new century method right here, urbanization stop, the answer is not really. If you look at it here, if you're familiar with Kansas City, to the north to south or south part of it, especially Johnson County and Jackson County, in the two sides of the state, two states, you definitely see the further development there. So it is definitely non-stopping. All right, and here is something that we have been doing for the Kansas City area. As I want to tell you, this one tells you the up panel tells you what's going on for the different type of land uses. For example, here, the yellow, orange, and the red tells you the urban peak source, which means okay, also building over there, right? And here to quantify the urban heat island, here this sort of snapshot tells you with that setup. Okay, this tells you the model simulations. Okay, you over here, the urban core area corresponding to the flat middle over here, you see the temperature almost 30 degrees, which is almost like more than 90 degrees. Well, in some rural areas, for example, it's one in rural area, actually it's down probably about 22 degrees Celsius. That's corresponding to almost 15 to 20 degrees Fahrenheit difference. This is how we utilize the downscaling technique to tell you what's going on for the urban heat distribution, right? And here we can also do some other kind of stuff. For example, here we are now looking at the present day urban heat source distribution. Well, this tells you about nearly 70 or 80 years ago. We did see the differences, right? We still have the Kansas City metro area, but it's much smaller and in intensity is really reduced. And then we are able to tell using simulation to try to kind of mimic what's going on if we're going to have some urban peaks like this one. And then we can also quantify the urban heat island effect. Let's give it this one. All right, another utilization of the downscaling study, we are also able to look at some kind of mitigation strategies. We know the urban area is really hot, and then there's one strategy is suitable cool roof material. The idea is that here you're going to see this kind of regular roof. You're going to also more radiator simply because it's really dark. Well, if you replace them with some kind of cool roof, right, with a white color, reflect more solar radiation. And then, of course, in reality, we cannot really do that, right? We are not able to ask everyone to change your roof. But in the modeling world, we can do it easily. We can simply just tune a parameter called albedo and then run 2 different simulations, then tells you quantify the result difference. That will give you some kind of mitigation potentials, right? So this is kind of mitigation strategy and look at it here. And basically, when we look at this kind of the cool roof material replacing the traditional roof material, we are able to look at it here. And the surface air temperature, it can reduce almost half degree to about 1 degree Celsius. While the surface skin temperature, the change is huge. It could as large as probably 2 to 3 degrees Celsius. It tells you mitigation definitely counts. Okay. All right, I think probably it's a good idea for me to stop over here, even though I still have a couple slides. I have to say over here, try to get this in. All right, thank you. Question? Yeah, please.
Forests are very diverse, and there's been some research showing the height of the forest, the density of trees, cools the earth below. Is that incorporated in the dams?
Yeah, that's the question. Yes, when we look at the different, land use, land cover trees, we definitely have different part of it, and you different kind of landscapes. In terms of trees, actually, we have different types of trees and different kind of crops, shrubland, and we are able to utilize something kind of remote sense or satellite data set. Tells you what this pixel is, the majority of those kind of trees. And then we are able to prescribe even model to tell you there might be some kind of difference between the app. And I'm happy to talk about the trees. Actually, trees is another very important mitigation scenario, mitigation strategy to reduce urban heat as well. All right. One question? I have 2 or 3 minutes. Yes, please.
Thank you for this talk. I honestly, as someone who studies the environment, I'm anxious all the time about climate change. And, I'm just like, okay, I have like 6 years before everything goes to crap. So I'm just wondering how you, as someone who studies climate change in depth, kind of deal with the anxiety that we, in my generation, experience all the time.
Yeah, I know, I'm teaching a class called climate change impact assessment and policy response. A lot of students in the class, they kind of show that, hey, Dr. Stein, you always talk about negative things in your class, right? Make us really anxious, right? That's a really bad thing, but however, we need also to be optimistic as well. Nowadays, with very, very, different tools. For example, we do have observations, including the kind of in-situ observations, for example, those kind of sensors that you put over here in this facility. Across the whole world, there's also satellite image, there's also remote sensing, there's also drones, right? UAVs, lots of information we are able to get. And first of all, we want to persuade people it's real. It's not really something really in the movie, Hollywood movie, right? It's happening. Actually, what happened during the past few weeks, they definitely took kids, right? What's going on for the heat dome in Southwest? What's going on to the northeast of the flooding events, right? So that's definitely something that we want people to know. However, I want to have another opinion that you cannot really always blame some weather extreme into climate change. When we talk about climate change, at the beginning we showed here, we need to look at the really long-term data set, right? We see the trend. However, if you go back to the picture that we showed, some years we definitely see kind of decreasing trend. So it's going to be combination of the human activities together with some natural variabilities. We cannot really always trying to ask you, hey, there's a heat over here, there's flooding, there's a drought. It's always because of climate change. It's true. We want to get people's attention, but we need to be scientifically, try to be fair, right? We cannot really mix climate change with some kind of 1 or 2 pieces of weather activities. Hopefully that will let you feel a little bit better. A little bit, I know.
Thanks to Ben Bang and all of our speakers thus far. We have a break, 30-minute break. Keaton Cameron is going to be bringing out some beverages and snacks. Please leave this room so that we can cool it off a little bit while we're out elsewhere. It's also cool in that other part of the building. So we can wait for 30 minutes.
Yeah.
Is that the, now up and allowed? Yeah. Yes, they got.
Everything is in order. We're ready to run. I'm very excited to welcome Gretel Keeper here from the Chicago Botanic Garden, and I'll share a little bit about her background. So Gretel Keeper manages the Plants of Concern program, which is a community science rare plant monitoring program at the Chicago Botanic Garden. Gretel has a background in research on native plant species, having worked with the Echinacea Project based at the Chicago Botanic Garden and with the Nature Conservancy in Minnesota, studying the effects of management practices on the threatened Western Prairie Fringe orchid. Yeah, ooh. You might even see some pictures. She currently serves on the board of the Illinois Native Plant Society. And these are Gretel's words, not mine. A long time ago, Gretel studied art and biology at Grinnell College. And then, yeah, Susan also went there. And then went on to get an MS in plant conservation and restoration from the University of Wisconsin-Madison. She learned to ID plants during her first job out of college at the University of Minnesota's Cedar Creek Natural History area. So I'm excited to hear about community science and rare plant monitoring.
Okay, I'm gonna figure this out. Sorry, technical difficulties. You're all good. Okay, can you hear me all right? No. Oh. You can turn it on. Is that okay? Okay. Cool. All right. Thank you, Erin, and thank you for inviting me to speak today. I'm excited to be here and be part of the Tyson community for the day at least. As Erin mentioned, I'm going to talk about community science. Yeah. Oh, yeah. Okay. Wait, I'm gonna toggle it over. Yes, yeah, great. Okay, so Plants of Concern is a community science rare plant monitoring program, and no surprise, I'm going to talk about community science and rare plant monitoring. And so I'm going to figure out first how to advance my slides. Is it the clicker or no? Sorry. Oh gosh, darn it. Okay, just click. Oh, perfect. Okay, so Plants of Concern has been active in northeastern Illinois for a long time, since 2001 when it was started at the Chicago Botanic Garden. We engaged volunteers there since then, and in 2021 we moved to start a program in southern Illinois. just to give you a sense of the scope of the program, our numbers, in 2022 for these regions combined, we had 228 community scientists active at 314 sites. They submitted over 1,000 reports on over 260 rare plant species. So a lot of data coming in. Oh, we're bouncing up and down. So I like to think about the three elements of the program: people, plants, and partners. And I almost titled my talk "Matchmaking for Plant Conservation" because really, it's all about connecting people and plants to support healthy humans and habitats. But we're not only connecting people to plants, but plants to people and people to people and partners to plants. And what I missed, people partners, all of these combinations. The people are community scientists. These are volunteers, trained volunteers, land managers, researchers, advocates, and artists. We're looking at rare, threatened, and endangered species, the local and regional trends for these species, invasive species that are in the vicinity of our rare plants, and we learn about the distribution of rare plants and the habitats that they require. And that's part of our partners are researchers. We share data with land managers, and we're just starting to get into facilitating this sharing of plants and seeds between partners, which is really exciting to me. So you probably are familiar with iNaturalist as a community science platform. I know I got to see the observations that are coming in for Tyson on your website, which is really cool. That's an example of an opportunistic or unstructured data collection method for community science, where basically anyone can take the opportunity to make an observation at any time and submit that observation. Plants of Concern is a little different. We're a structured community science program. So basically we are required, we rely on trained volunteers, and those folks go out to a particular location where we know there's a location of a rare plant, and they visit it at a particular time when it's usually in flower or most visible, and they go through a very set protocol at the location to record information about the species and the location that it occurs. So in that way, we can collect very specific standardized data. In order to do this, we're lucky to have a very advanced tech platform. Right before COVID, as COVID hit, we had our last in-person training, and now we rely on online training to train volunteers. And this is a series of 6 video modules and quizzes that takes about an hour and a half to complete. So in that training, we teach about what are rare plants, why they are rare, and so rare plants differ in why they are rare. Sometimes we just have small population sizes. Other times you can have large populations, but very few populations. They're often limited by habitat that they require and resources, and often face reproductive challenges. We talk about threats to native species, which you all heard about in other talks already today: loss of habitat, climate change, invasive species encroachment, loss of connectivity or fragmentation. and disruption of natural ecological dynamics like described fire. So that's kind of painting the doom and gloom for rare plants, but we put a positive spin on it because community scientists are making a difference in the data they collect. So we talk about why we need community scientists, how their data are important, and how monitoring helps save plants. After after the online training, we invite people to come out into the field with us, because that's really where the magic happens, and you can't become sort of confident in the field until you've gone out and monitored. So we we'll hold these field training events for new volunteers, and we usually locate these in areas where we're looking for a new participant to take on an assignment that's open, or a place where we have an experienced volunteer that is looking for more help. So that pairing of experienced and inexperienced volunteers is a great way to get people on board. We have a website where we have a lot of like public facing information about rare species. You can search up all the listed species in Illinois and learn about how to identify them, their bloom times, and other species information. We also have an accounts side to our website, so land managers can log in and see all the rare species that occur at their sites. And look at all the reports that have been submitted over the years for each rare species. And we have a great data download feature for community scientists and land managers, where they can download the report data in PDF or spreadsheet format, or also as a spatial file for loading into GIS. Also really important, rare plants information and location is kept confidential. So user is only able to see the information for the assignment that they, for the species and location that they are assigned. And that's really important for like keeping trust with the land managers that we work with. We were lucky to get funding to also make a custom mobile app to collect data. So instead of carrying a GPS and a clipboard and a data paper form to fill out, we now are able to navigate to a rare plant location, walk through the protocol that we use, collect new GPS information if the distribution of rare plant has changed, and submit that in real time, which is really efficient and a great asset to our program. So I talked about the impact that community scientists have on plants. And the reverse is true. Plants impact people. And the program, I think this is a quote by Kathleen Garnis talks about that. Plants of Concern has transformed my life, pulling together all my abilities and interests toward a higher purpose and fuller community. It has given me new eyes to see things more carefully, appreciate nature in all its diversity, and communicate that with others. The physical exercise involved in monitoring stewardship keeps me healthy and young. Kathy is a longtime community scientist and also an amazing artist who is focused on orchids. So the rare plants, 1/3 of all plants may be at risk of extinction, and so why rare plants? Not only do people get really excited about going out in the field and seeing things that aren't common, rare plants also can play very important roles in networks. This figure shows an example of that. Kate Havens at the Botanic Garden looked at the plant insect visitor community in the student system, and the federally threatened Cercium pitcheri wood thistle, you can see kind of plays a very important role in insect visitation. So in this system, without that plant, there would be huge ramifications for the rest of the community. So I mentioned that we focus on threatened and endangered species, ones that are either federally or state listed. And we also can really monitor any plant that a land manager is interested in. So, for example, in northeastern Illinois, pawpaw is not common like it is here. So we have land managers in northeastern Illinois that are monitoring pawpaw. So the data that we collect, we annually visit a location, collect GPS data, we count the number of individuals of the rare species that we're looking at and know whether they're flowering or not flowering to get an idea of the reproduction. We record the most abundant native species associates and also what invasive species are present in the vicinity of the rare plants, and then like percent cover of those invasive species. Other impacts that we look at include like deer browse or unauthorized trails and things that we can report back to land managers to let them know are important to consider. We also take note of any evidence of management that has occurred in that affects the rare plant population, like evidence of prescribed fire or woody invasive removal or piles of herbaceous plants that have been pulled, and record that. And we can also submit photographs and make just general notes about what we see in the field through the mobile app. An example of the data, this is trend data for Cypripedium candidum, the small white lady slipper, in the forest preserves of Cook County, populations that have over 5 years of data. And the pink line shows the generalized linear model trend line. You can see it's increasing. The population size on average is increasing over time, and data like these helped move. Sorry, it's a mountain. Helped move this species. It was listed as a threatened species back in 2004 on the Illinois endangered species list, and now because of data from plants of concern, actually been moved off of the threatened and endangered list, which is really exciting to see. So the data that we collect also gets used to inform management at sites. Land managers use the data to guide invasive species removal. Basically, we are more eyes on the ground. Our community scientists report things back to land managers so they can prioritize land management and use their resources wisely. And I was happy rare species was helping part of that. Because it's not just about the rare species. They're in a community in the system, and I really feel like we're focusing on rare plants for the benefit of all. We also send data to the Illinois Natural Heritage Database. This database collects all of the rare species occurrences in Illinois and then feeds the data into NatureServe, which is more of a global database for rare plant occurrences. Other rare. We partner with researchers. Shout out to Raphael, who is a PhD student in the Northwestern and Chicago Botanic Gardens Joint Conservation, Plant Biology and Conservation Program. He studies how genetic augmentation can help recover rare species and improve conservation outcomes. Basically, this works like small isolated populations of rare plants can decrease over time and eventually become extirpated if nothing's done. So in cases like this, we're advocating for experimental plots that introduce more genetic diversity by bringing in seeds or plant copibules from nearby populations and establishing the experimental plot next to declining wild populations to result in a more diverse, healthy, reproducing population. And when I talked about kind of matching partners in plants of concern, like we're because we work with so many land managers, we were able to know who has what species and how they're doing and make these connections so that trading seeds between sites that can happen, which is sort of like pushing the envelope of plant conservation right now. It doesn't seem like it, but it's really not done very often, but it's starting to happen, which is really exciting. A case study, the Dune willow. This is Salix certicola. It only occurs in 2 counties, lakeshore counties in Illinois. It's state endangered and a dune habitat specialist. There are only or fewer than 20 individuals left in Illinois, and got an interesting biology. It's a dioecious species, so it's separate female and male plants that may be part of the reason why it's not doing so well, but basically being a dune plant, it's been impacted by lakeshore erosion. In 2020, we had historically high Lake Michigan levels, and this is what Plants of Concern monitors David Johannesson and Karen Lustig saw. These are dune willow individuals popping up in the flooded soils, interdunal soils. So they kind of raised the alert that something needed to be done for the species. And willows grow very well from cuttings, so we were able to take just branches, divide them up into, I should say, folks in the propagation department at the Chicago Botanic Garden took little cuttings and basically just produced more plants for us, which we, from just a few cuttings from representing all of the individuals that we could take cuttings from, we planted back at Illinois Beach State Park where this study is going on, over 200 plants over the course of the last few years, which is really exciting. So we created these experimental plots near the existing rented plants. With the help of volunteers coming out and doing a planting, we GPS every one of the plants, and unfortunately, we've had some drought conditions which have led to a lot of mortality of the individuals that we planted, but just this spring, we were able to collect seed from 2 female plants. And my fingers are crossed that germination is in progress at the Botanic Garden. So we'll, you know, if we have successful germination, we will have more genetic variation to plant back at the site, which is really exciting. I'd just like to acknowledge that we rely on a lot of funding sources. There are our funding partners. And I got an email yesterday, so I should put another box here. The Illinois Department of Natural Resources is gave us some funding, so that's really exciting. That's not just me. We have a lot of staff. Note TBD program assistant, if anyone is interested, submit your application. And thank you, community scientists and artists and photographers who donated their artwork. And shout out anyone who is interested in monitoring rare plants. Our tools are available for starting other rare plant monitoring programs. So Tyson, if you're interested in setting up rare plant monitoring, I'd be happy to help you do that. You know, we've got all the technology available to share. So that would be really exciting. Thank you.
Visual monitoring at Belleville Prairie, and what have your participants think of it, if they were?
I wish we had been monitoring at Belleville Prairie. Yeah, it's outside of our northeastern region, but I would like to think that if there were more eyes on the ground there, and that the outcome would have been different. Yeah.
So in terms of your volunteers, where do most of them come from? Are these like visitors of the gardens? I'm just kind of curious about how you recruit. And then also with that, have you seen a difference, like kind of pre and post COVID with going digital for some of your training? Do you see like more participants, or has that changed at all? I don't know if you've looked at that.
Yeah, good questions. Okay, so I didn't talk about it, but our community scientists are typically never come to the Chicago Botanic Garden. They're often already active as volunteers at forest reserves or park districts. They're usually already interested in native plants, maybe members of the Illinois Native Plant Society. So we see a lot of crossover between so the Chicago region has like a rich history of volunteer restoration. So often people that are doing stewardship restoration work at sites see the fruits of their labor and get interested in rare plants and start monitoring them. Or we have people that are submitting, already observing rare plants and submitting data who see trends going down and then want to do restoration work and start volunteering as restoration stewards. And so we have more people interested in becoming community scientists volunteers than we can match with vocations pretty much. And that's sort of the bottleneck of our program. is making that match between a suitable assignment for someone that's, you know, not super far from where they live. And so that's kind of a tricky aspect of the word. Yeah. Anything else? Other questions? All right, thank you very much.
Hello, everyone. Our next speaker is Dr. Emily Reed. She's coming to us from Virginia Tech, where she's currently a postdoctoral associate with the invasive species working group, where she does a lot of research on science policy of invasive species. Before that, she got her PhD in ecology and evolutionary biology at North Carolina State University, where she studied the landscape genomics of the invasive tiger mosquito Aedes albopictus. And before that, which I think is really cool, is that Emily got her undergraduate degree in French from UNC Asheville. So without further ado, take it away, Emily. All right, let me see if I can get this thing set up. All right. Oh.
Up the counter.
Yeah. I'm holding it. Is that all right, everybody?
Yeah.
Cool. Thank you, Lauren, so much for that introduction. Thank you all. This has been really cool to be here. It feels like a unique place. Not that I know that I have a ton of experience, because like Lauren said, I was a French major and undergrad. But kind of on that note, out of curiosity, how many of you did field work or had some kind of outdoor research experience like before or during undergrad? Okay. How many of you like spent a lot of time outdoors as a kid? Like would say you're kind of that kid. Okay, so I think I'm getting like a little over 50%. So this is my backyard now. When I was a kid, I did not go outside. I lived in a beautiful place with lots of woods. I was very lucky that I did not go outside. And there were two big reasons. One was allergies, and the second was mosquitoes. React horribly, don't like them, did not enjoy it. So of course, what did I do? Got my undergraduate degree in French, took a few years off, and somehow ended up in a PhD program studying mosquitoes, specifically Aedes albopictus, probably the worst human biter in the Southeast US and a lot of parts of the US, the most invasive species, mosquito species worldwide. And when I say that, I really mean it. Like it is everywhere. It is ubiquitous in human environments, especially in these like residential places where we all are, where we like to congregate, where we like to spend our time. And when we're thinking about, you know, getting people into the outdoors, they're really where we're trying to get people out into. So they're not great. But as I was learning about this very annoying and also quite deadly mosquito, I started investigating and getting really interested in why it was so good at what it does. So Aedes albopictus, the tiger mosquito, like other mosquitoes in its genus Aedes, is a container breeding mosquito.
And in its native habitat, those containers are generally tree holes. And there's a great reason for this. So for these mosquitoes to actually hatch from their eggs, they have to go through a desiccation cycle. So the adult female lays her eggs in a body of water, and the eggs actually have to completely dry up and then be re-inundated with water before they hatch. And that is for predator avoidance. There are lots of things that will eat the aquatic larvae, but those predators are also aquatic. So if you lay your eggs somewhere and it dries up, then when they hatch later, there's not going to be a lot of predators. Well, guess what? Turns out that makes them really, really good invaders. Makes them really, really suited to live among humans. And we've seen this. So they were introduced to the US. They're first detected in the 1980s. We had them where I grew up in North Carolina by 1992, and they have just rapidly expanded. A lot of invasive species, we talked about this kind of like cryptic stage where they're there, but they're not being detected. The cryptic stage for these guys is quite short. They're expanding in Europe, they're everywhere. And they're everywhere as adults, they're everywhere as eggs and larvae. This was one of my favorite studies, really motivated my research from 2017, where it's a citizen science project in Portugal, where they had people take photos of mosquitoes in their cars. Many studies have looked at all the different breeding habitats because they're container mosquitoes. So they went from tree holes to our refuse in our yards, to our pots, to our trash, anything that can hold water. And so, you know, again, kind of still coming from this place where I just really did not like these guys. I wanted to know how they got here and understand not only how to get rid of them, but how that works, how they, you know, disperse across these systems, because these guys, they're not very good at flying. They're not really good on their own at dispersing. Most adult mosquitoes only disperse about maybe 100 meters as adults in their lifetimes, but they're everywhere. So they can go from here to our cities. And again, they're super successful invaders. And again, French major, not taken a genetics class in my life until graduate school. We decided we were going to investigate this using population genomics and really looking at a lens of invasion genomics. So with invasive species, we usually think about a small population being introduced into a new place. There's pretty low genetic diversity because they're just a sample of that native range. But in urban systems where a lot of introductions happen, you can get multiple introductions either from the same place or different places, and that can really increase genetic diversity. And so here's one representation of that. And more genetic diversity generally increases maybe the likelihood that they become established and spread, and also the potential for them to further evolve and adapt to the new environment, their non-native environment. One thing that's also really cool to me is thinking about how if they're already adapted to live among people, how that can facilitate gene flow and connectivity, and they can be even more successful and have a greater potential to evolve. And, you know, there's some evidence already that that's what's happening with Aedes albopictus. A lot, or some, I might say a lot of that evidence comes from some of the people in this room. So I wanted to investigate what was allowing these mosquitoes to be so successful and spread and genetically connected at a pretty fine scale, which kind of leads me to my research question that I'll talk about today, which is to investigate these patterns of genetic connectivity in Aedes albopictus and identify landscape features that influence that gene flow, that connectivity. So the first thing we had to do is we needed to think about where we could do this. So this is from the CDC in 2018, which is when I conducted my sampling.
And these were, these kind of green, yellow regions are where they're most likely to reproduce. I was at NC State, so in North Carolina, we did a kind of thought exercise, cost benefit analysis of where we should do our sampling. Well, we had no money. So it turns out the best place to do it was in our backyard. So that was Wake County, North Carolina. And actually, it is a pretty interesting place. It was in between 2012 and 2020, one of the top 5 fastest growing counties in the US that already had a population over 1 million. There's that caveat that makes you really, but it was already a large county, had a large population, and it continues to grow. And what you see here is an example of not an urban-rural gradient so much as an urban-rural mosaic. So this is kind of the development level. So the darker red is like more intense impervious surfaces, high-rises buildings, and then kind of the lighter pinkish are things like neighborhoods, maybe a little bit more residential. And one of the first things I learned is I wanted to do this nice urban-rural gradient study is that places in the Southeast, that's not so simple. It's really suburban sprawl. And suburbs look different, rural areas look different, which it complicates things, but also Aedes albopictus like to be where humans are and where they live. So I decided that what I wanted to do was just kind of randomly sample this county. So I had 60 sites. I ended up using 39 of them in my study, and I sampled adult Aedes albopictus mosquitoes. And I don't know that these pictures really do justice, but we did have like a great range of sites from, I think my furthest one was about a half a mile from a church parking lot that I kind of tromped through the woods to things like right in downtown. And again, we wanted to take this and look at genetic connectivity. So I'm going to very, very briefly touch on the genetic component, because one thing that I knew going into this was that Aedes albopictus does not have a lot of genetic structure. So kind of a way to think about this is if any of you have done 23andMe, it'll try to tell you where you're from. Is it very good at it? No, but it can give you some information. When we look at population genetics of some other mosquitoes like Aedes aegypti, very closely related species, we can at least tell where they're coming from if there's a new introduction invasion. Turns out we can't do that with albopictus. It's super cosmopolitan, it's everywhere, it's spread quickly. So in order to really look at any patterns, especially at this fine a scale, we really needed to kind of like up the ante. So we used genomic techniques where we looked across thousands and thousands and thousands of locations in their genome to detect these single nucleotide polymorphisms. So just one change in their genetic code. So in total for this study, I had 256 mosquitoes, 39 sites, and about 4,000 different variations in genetic code that I was using to try to see if I could tell how landscape was influencing how they were moving, how they were genetically related to each other. So the first question was if it was just a matter of distance. The further apart you are, the more genetically different you are. That's not the case. So this is a little much, but here's some like sample sites. This was from like a preliminary data collection we did. And you can see kind of the yellow and the green point and where they are in proximity to each other. On your left is where they are in space. On the right is how genetically different they were in their genomes. So distance did not correlate with genetics. So how do we look at what influences how these mosquitoes are related to each other? So I use this tool called gravity models. Like Dr. Murphy, I had wanted to do this and had put it off until I got this invitation and I said, now's my chance. So these gravity models are really cool. They use like graph theory. They're generally used in like resource economics, trade, but they've started to be applied to genetics and ecological questions. And what they basically do is they take both the landscape between sampling points and the features at a sampling point and use that to explain the variation that you see, in this case, in the genetics. And so to kind of show you what that could look like in terms of landscape and genetics, so you could have something happening between these sites that affects connectivity. In this case, this is a representation of a river. The mosquitoes can't cross the river, so we don't see connectivity directly between sites that are separated. One of the really cool things is you can integrate that with what's happening at the site. So this could be something like habitat suitability or even habitat size, and saying, okay, like larger habitat, better habitat, they're sending more migrants over, so they have more immigrants going to other locations. You put this all together. A big caveat with this approach is that you can't just throw all of the data that you have at it and see what comes out. You need to have some hypotheses about why site-specific variables and between site variables would be impacting gene flow. So hypothesis driven. And so we had a set of site-based variables and between site variables that were somewhat similar. So at a habitat point, we looked at the mean percent impervious surface. So, you know, again, they need water that dries out, but it has to have a place that can collect water. So impervious surface is related to habitat availability for their larvae, as well as the proportion of forest cover, the distance to the nearest major road. So that was inspired by a paper from Dr. Medley, as well as others that have followed, and then housing density. So how many residential properties were within this habitat where we collected the mosquitoes. Between sites, we again looked at the amount of impervious surface. We looked at the proportion of forest cover. I showed you that map earlier of the different development types. So we looked at the proportion of each of those 4 development types. And then we also did like a least cost paths, looking at the distance along highways between sites. And so we had all of those. And then we, as well as just the straight distance between sites. And we looked at how well those different elements correlated with genetic connectivity between all the sites. So again, correlative. We did some model selection to identify which of these variables were most important. And it actually ended up being really nice because using Nay's genetic distance to try to ward off the genetic population geneticists who might be asking, we found that there were 2 site-based variables and 2 between site variables that were created the best model to predict genetic connectivity. So the first, they're both related. Again, it turned out quite nice. I was shocked because that had not happened before. So we had 2 elements related to roads. At the site, it was the distance to the major road. The further away mosquitoes were from a major road, the less genetically connected they were with all the other sites. And then between sites, the longer distance there was along the highways to other sites, the more or the less genetic connectivity. So you had to go further along roads, you're less genetically related. Second part, these are also kind of related. The first I'm going to talk about is percent forest cover. So the more forest cover there was at a site, the higher or the more genetically connected the mosquitoes were with other sites. We haven't totally dived into this, but this does seem to be somewhat parabolic. There does seem to need to be some element of like human residentialness. But overall, this was the trend that we saw. And then what's kind of interesting is that open development, which is going to be like managed greenland, things like big open yards and golf courses. So the more of that there was between two sites, the less genetically connected they were. Oops. So overall, what we saw was that these road networks and these wooded environments appear to facilitate gene flow, of course with caveats. So this is a very fine spatial scale, but it's a spatial scale that's relevant to management, especially in North Carolina. They don't have like district mosquito control units. Counties are responsible for monitoring for mosquitoes and kind of like handing down rules to private mosquito control businesses. And again, it's super hard to tell genetically how different these mosquitoes are because it's so chaotic, they're so cosmopolitan. But the fact that we found things that were statistically significant is kind of exciting in and of itself. And it aligns with what previous studies have shown. And to me, what's really cool about it is it kind of reflects back on that story. You know, it's like they're cosmopolitan, they like people, they spread through roads, but there's still these container mosquitoes. They still started like in the trees. This might be just too much storytelling, but it's kind of cool. Like they still need that element. And that those came out as like the most important factors at this scale is interesting to me and somewhat unexpected, and it has motivated me to want to continue exploring these things. And you know, right now in my postdoc, I've moved away from mosquitoes, as Lauren said, but I really appreciated working on this presentation, and I wanted to give a huge shout out to my PhD lab, Conservation Currents. There's our lab, as well as my funders during my PhD, and also the groups that I'm with now at Virginia Tech. The Invasive Species Working Group has really helped me think about these questions in different ways and think about invasive species and our interactions with them differently. And I don't know, it's been really nice to revisit this. So again, I appreciate the opportunity. I'd be happy to take your questions.
So across different mosquito species, what techniques do they use besides container breeding to spread?
So the question is like in other mosquito species, what is their dispersal mechanism? So the container breeding, the reason that they're so successful at spreading that way is because of us humans, because we like to move stuff around. So the reason that Aedes albopictus was introduced globally is because of the tire trade. So tires are stored outdoors. They're really good at holding water, and the eggs can stay in that desiccated, like dried up state for quite a while and still be viable. So in their native range, tires were being built and shipped and again, stored outdoors. So they were, built and stored in their native range. They laid their eggs, and then those tires were shipped to the United States and other places. They got there, they were stored outdoors. It rained, the mosquitoes hatched. Other species that don't use containers, some of them are much better dispersers as adults. So anopheles can disperse much further on their own. But actually I'm not sure about like the main dispersal pathways for other genera. Like there are some other genera that do use containers.
But I mean, they're great at living around people and they reproduce very quickly. And they're adaptable. But again, I think Aedes albopictus just does it best. Yeah.
You mentioned you didn't go outside because you hate mosquitoes. Now you study mosquitoes. What do you do when you go outside?
I wear a lot of DEET.
We have a question from our YouTube channel from Jonathan Losos. He says, if I heard you right, you had about 9 mosquitoes per site. Are there any guidelines about what sort of sample size one would want for a study like this?
Well, first, John, I think that one of my coworkers knows you, so David H says hi. That's a great question. So yes, so there have been several, more methodological papers looking at the number of individuals you need for the kind of genetic approach we were using, which is called reduced representation genomic sampling, so that you can have lots and lots and lots of genetic markers, and you don't need as many individuals to find differences. So I actually had less than 9, I had about 6 to 8 individuals per site. And that is on the low end of what they recommend to be able to understand, truly capture the genetic variation at a site. So I didn't talk about it here, but we did look at genetic diversity within a site and more traditional PopGen approaches to genetic structure. And we did find that broad genetic diversity was correlated with the number of individuals we used. So then we did fancy things, not really that fancy, rarefaction, where we look at kind of the genetic diversity when we only take a few of those mosquitoes at sites where we had more and adjusted for that. And we found that it did not significantly change our genetic connectivity measure that was used here. So even when we only sampled a few mosquitoes from sites where we had more samples, we kind of got the same result, the same picture. Yeah, ideally, we could sample everywhere and lots of individuals, but that was the trade-off that we decided. And I don't know if it was the right choice, but I think we can justify it a little bit, and hopefully some of you all will do different things and we can see, and compare. Yes.
So, I've been told over and over again, Alopecia has no genetic structure, right? Kim found evidence, you found evidence. My question for you is, do you think the smaller scale is really the scale that maybe the structuring is happening versus looking at a wider geographic range? Or do you think the molecular techniques, the genomics techniques are better and that's what allowed you to capture it? Or are these people just wrong?
So they're definitely not wrong. I mean, it's a hard species to work with. It was super frustrating. I think that when you look at larger spatial scales, one of the big things is the within population, within site variation versus the between site variation. So, imagine having 2 bags of different colored balls and there's 3 different colors they can choose. If one bag is all blue balls and one bag is all red balls, you're like, oh, they're different. But what if one bag is 6 red balls and 4 blue balls and the other, oh shoot. Anyways, you know what I'm trying to say. So it seems like at a finer spatial scale, because we can get these more fine landscape variables, we have a lot that we can look at in terms of explaining those little differences. I think that helps us find these things. And obviously, the more balls that you have, then the more variation you can detect. But it's hard. I mean, we're talking about, I'm explaining you know, I know Dr. Murphy was saying like, oh, my R squared is so small, it's like 0.4. I wish I could get something like that. I heard that very, very small differences. But for within the population genomics world, it's still important. Yeah, so I think it's a little bit of everything, but I do think scale is quite important. Sorry for that weird analogy. Thank you all.
Thanks. I'm very excited to introduce our next speaker because networking and knowing people works, y'all. So it's very exciting to be able to invite someone to our community who I met virtually. Thank you, Amanda. Because I know, right? Because of all the webinars, right? And all the virtual conferences and all those kinds of things that have happened. And so I was able to see ESB, and I want to say that it was a UFERN conference moment. So UFERN, Undergraduate Field Experience Research Network. There are more people like me at other places that are very interested in how to promote diversity, equity, and inclusion in these spaces where we have research experiences for undergraduates, and in our case, of course, also for our high school folks as well. So anyway, that networking works, y'all, so it's great. So it is my pleasure to introduce Dr. Nia Morales, as she's an assistant professor of human dimensions of wildlife. That's a thing. In the University of Florida's Wildlife Ecology and Conservation Department, where her teaching and research generally focus on the role of social science in the conservation of fish and wildlife, as well as issues of diversity, equity, and inclusion in conservation. Dr. Morales received her bachelor's and PhD from the University of Florida in Wildlife Ecology and Conservation, and also has a master's of environmental education from Florida Atlantic University. Previous to her faculty role, Dr. Morales was the head of the Center for Conservation Social Science Research within the Florida Fish and Wildlife Conservation Commission's Fish and Wildlife Research Institute, where he led a team of social scientists that provided support for the development and implementation of social science inquiry that helps the commission better understand the complex relationships between humans and the management of natural resources. I'm very excited. She has a very recent publication that's come out that I believe that a lot of this talk is about, and is very interested in pushing, I don't know, boundaries, breaking down traditional ways of thinking about things. And so I'm very excited to hear what you have to share today. So please help me in welcoming. And the most difficult part of everything is about us. It's
gonna help.
Thank you. Okay. Yeah, thanks for inviting me. This is a really interesting place, and I feel like there's a really great sense of community here. So my talk is very different from all the ones that you've seen earlier today. And it's funny because when Susan invited me and I kind of told her about the types of things that I do, I was like, I can talk about X or Y, and she goes, all of it. So that's one of them. So one of the things that I look at
in my research, in addition to looking at undergraduate field experiences, is kind of looking at the entire field of conservation, how we got here, and what it means for the type of work that we all do, and how we can change it to make a better future. Enter, down, up, down,
Or over.
over. There's not very many directions left. Like that way. Okay.
Did I go too long?
Yes, that's good. And I do also I'm gonna go back because now I know how to do this. I also want to mention that the research that I'm engaged in now comes out of my own personal experience. I never really acknowledged that I was typically one of the few women in the room or one of the only brown people in the room until much later in life. And I started wondering like what that means for me as a professional. And it's really become something that sort of pushed the type of research that I do to create spaces that are much more inclusive so that people don't have to feel like, why am I the only one? But that's just an aside. So we're gonna do a little bit of a history lesson first. So the idea of conservation in the West specifically is actually a descendant or antecedent of what's called terra nullius. Has anybody ever heard of the term terra nullius before? So that's an idea that basically before Westerners got to a place, nothing existed. And this is related to also what's called the doctrine of discovery, which was a document from the Catholic Church that basically said anywhere there were not Christians, you can colonize. And that was the idea that any people that lived there were less than. So they weren't Christians, they weren't part of the European sort of culture. Therefore, they were open and right to be colonized. And this actually became the basis of how we think about conservation. It seems like a big jump, but essentially in the Western, well, in the United States and Canada, pretty much, when we started building the conservation sort of field, the idea was that any place where we go, the people who live there are not civilized. They don't know how to take care of their land. So therefore, we, being scientists or being people who are better educated, have the right to tell them what to do, kick them off the land because we know better. So that led, the concept of terra nullius led to the idea of fortress conservation. Has anybody ever heard that term before? So fortress conservation is the idea that in order to protect natural spaces, we need to have essentially a fortress around it. And that the humans that are there are doing negative things to the environment. So we kick them all off, we preserve the land, we've made a benefit to conservation. In my particular field of wildlife conservation, we have a very specific version of this that came about sort of in the mid-1800s. And they actually specifically call fortress conservation the Yosemite model. So as we built our national parks and kicked off all the Native Americans who lived on the land, that model became what we have essentially exported to other countries. And it's caused a lot of issues across the globe with indigenous populations who may have lived and worked and gained livelihoods from these areas that now they don't have access to. So in US and Canada, there's something called the North American Model of Wildlife Conservation. Has anybody heard of that? A couple of hands. So the North American model is the dominant model or philosophy of conservation. If you go to a wildlife ecology undergraduate program, you will learn about this. This is what we teach as the basis of how we think about the philosophy of doing conservation when it comes to specifically wildlife management. And it has its origins in the 19th century, and it's characterized by protected areas, restricting commercial exploitation of wildlife, having common laws and rules that are dedicated to species and habitat conservation, and also the federal and state, essentially government management of wildlife. For many people, this is the gold standard model of conservation. And while I do agree that it has benefits, I do also think that in order to move forward, we need to critique the past.
So the North American model was inspired partially due to the criticism of elite sport hunters that immigrants and people from low socioeconomic status were ruining wildlife. So essentially, they believed that in their gentlemanly pursuit of sportsmanship, that other populations were hindering, causing the basically population declines of things like songbirds and other species. And because these were largely like wealthy, landed, educated white men, they had a lot of political power to basically set up the way that we think about conservation. So a lot of the early conservationists started a lot of the early colleges that offered wildlife degrees. And so this sort of philosophy was embedded in the way that we taught future conservationists about how to preserve wildlife. And so I'll go over the tenets of the North American model and sort of describe some of the critiques that have been pointed out about this model. And so it's a product of, white, mostly wealthy rural men whose ideologies really reflected the time that they came about. And it really wasn't exclusive to any other ideas of how the world should work. So the first principle of the North American model is that wildlife is a communal resource held in trust for the public by the government. Now, as written, this sounds fine. But if we know anything about the history of discrimination in this country, we know that not everybody has been able to participate in this process. And so our idea of conservation comes from this Anglo-European context and doesn't take into consideration non-Western ways of thinking, doesn't take into consideration other political frameworks. And so it basically excludes different ways of interacting with nature. The second principle of the North American model is the elimination of markets. So again, I mentioned that it came about because a lot of these wealthy sports people didn't want to see wild game sold. And so part of what they wanted to do was to make it more sportsmanlike. So here is a quote from, I can't remember what Hornaday's birth name is, but he was one of the leading conservation sort of big figures in the early 1900s. And he basically says, I'm quite certain that their experience with being songbirds is being caused by the slaughter of them for food in the north by the Italians and in the south by the Negroes. So clearly these were coming from elitist positions for people who were anti-immigrants and had racist worldviews. And they were blaming these populations for the declines of wildlife species. An allocation of wildlife by law. So this basically means that the government has the right to set up things like bag limits, hunting regulations, different regulations determining who can have what in what spaces. But this also misses the whole, it doesn't take into consideration the inequities and oppression that the US government has brought upon, for example, indigenous people. It doesn't take into consideration that laws have been meted out inequitably for a long time. And so different people had unequal access to these opportunities, and they also haven't been able to be involved in the lawmaking process. So principle 4, that wildlife should only be killed for a legitimate purpose. Again, so this was the idea that a true sportsman is the one who should be able to determine who has access to being able to shoot game. But what is a legitimate purpose? Who gets to define what legitimate take actually is? Well, a very small group of people did. And so this doesn't take into consideration traditional uses. It doesn't take into consideration maybe cultural uses, or even, it doesn't even take into consideration different views on how people relate to wildlife. This is saying that wildlife is a commodity, essentially. There are indigenous groups who have who have very different perspectives on the relationship between humans and nature, that humans and wildlife are essentially
on equal playing fields. Wildlife as an international resource. So again, this is one at face value. Yes, wildlife don't abide by political boundaries, for sure, we understand that. But it also doesn't really take into consideration that the laws and regulations of different areas are often determined by a very small group of people. So even within the United States, we often don't recognize the sovereign rights of indigenous groups. And so if animals are going back and forth between those groups, it's still the US government that takes precedent over how to manage. It's only recently that government agencies have been looking for a more co-management model where they're actually recognizing the sovereignty of tribes for their ability to manage wildlife who come onto their own lands. Principle 6, science should be the determining factor in allocating natural resources. So again, who's science? So we have always focused on the more European Western-based model of science without acknowledging indigenous ways of knowing, traditional knowledge, even local knowledge. It doesn't necessarily have to be from an indigenous group, but folks who have lived in an area for a long time generate a lot of local understanding about processes and about systems. But we don't always recognize that as science. We treat it as a little bit of an other. And then principle 7, again, as written, it sounds like a great principle. Every citizen has the opportunity under the law to hunt and fish in the US and Canada. But for, I would argue, hundreds of years, this hasn't, again, been meted out equitably. There have been times in US history where non-native or non-residents and immigrants from certain countries were barred from getting hunting licenses, were barred from owning firearms. So this idea that every citizen has an opportunity to participate in consumptive use is actually not something that has been equally accessible throughout our history. And it also ignores the idea that not everybody's relationship with the outdoors is consumptive based. So it prioritizes hunting and fishing over other types of recreation. So I teach an entire course in what I just told you in like 7 minutes. So I recognize that this also might not be your particular field. You know, wildlife ecology is the subset of ecology. But I kind of just wanted to introduce you to these ideas because what I'm interested in is how the future generation of scientists can learn what we've done in the past, take the good, take the problematic, synthesize that, and create a future where we are more inclusive in the way that we think about science, the science that we do, and the communities that we are engaged with. So in this paper that this talk is basically surrounded on, we posited a few ideas. But I do hope that, you know, maybe over dinner or whatever, if anybody wants to chat about this, it'd be nice to know like other things that you as future scientists and professionals think can help redefine conservation for centuries to come. So for one, seeing nature as an inclusive space. So historically, wilderness and outdoor areas have been a white normalized space. And natural spaces have also been a source of not just perceived violence, but actual violence. So from, you know, even though we often talk about civil rights as something that's happened in the past, we still have news articles like every year about a person of color being accosted in a natural space, right? So this is still an issue that faces communities. We still see, you know, stories on the news of people just from any marginalized background, LGBTQ people who can't be outside being their full selves. And so I think one of the first things to do is to acknowledge that the outdoors is a place for everybody and that everybody interacts with it in a different way. And the way that you interact with it is not necessarily right or wrong, unless you're being like a total tool. But you know, that there is opportunity for us to interact with the outdoors and natural spaces in ways that make us feel comfortable. Understanding inclusive relationships with nature. So it's really cool to see projects that, some of our other speakers have talked about that incorporate urban ecology. I think this is an area that has, within the past like 25 years has become a real focal point for a lot of ecology, innovation. Because we recognize that a lot of our population lives in urban areas, and in urban areas, there are habitats, there is wildlife, there is, there are systems in play that should be studied. And so I think understanding that, the study of conservation and conservation initiatives shouldn't just be restricted to like save the polar bears or, like things that have to do with traditional wild spaces, that they are also places where they could be in urban areas or suburban areas. And also to understand that it might be more engaging communities works better when it's something that they feel connected to. So instead of trying to get somebody to donate money to like Yosemite National Park, why not have people participating in like a citizen science event that's locally based, because then they can learn about the systems that are important to them and are still doing good conservation. Incorporating other ways of knowing. So again, not having a monopoly on what science is. I think this has been something that has arisen maybe in the past decade or so, when we're acknowledging that the way that we do science is exclusive, and incorporating local knowledge and indigenous perspectives actually helps us to be much more creative about conservation solutions. And it empowers these groups as well to have some agency in determining how they want to manage the natural resources that they. And one of the things that I wanted to bring to your attention is that I do think that there have been strides, and I think the first step is acknowledging the issue. I feel like they say that about, like Alcoholics Anonymous and stuff like that, first admit you have a problem. And I do think that, at least for what it's worth, the whole 2020 George Floyd thing did set off a new interest, renewed interest in exploring these issues. And that we have had some change. So for example, the Vatican formally rejected the doctrine of discovery this year. What good it does, I don't know. It's sort of like, sorry, we destroyed all of your cultures for the past 600 years. But I do think that it is a step in the right direction to acknowledge the hurt that has been traditionally caused, because I think it sends a message to these communities that you're open to having that discussion of what comes next. And I like to use, I know this is a giant chunk of text. There's a book called Cast by a woman named Isabel Wilkerson, and she has a really interesting analogy that I use in my class all the time. Essentially, the idea is that we live in an old house. None of us were here when that house was built, but we all live there now. And so it's got like some crumbling beams and plaster's not so great and stuff like that. But we have a decision. Do we fix the foundation, or do we just patch it up and move on about our lives and the next generation's problem? And so I like to think of it as this analogy, because I believe that if we understand how our house was built, we then have the tools to either decide to fix it or just patch it and move on. I hope that we actually fix the foundation and address all the issues in the like the bones of the house. But we also have to understand that we weren't here when it was built. So one of the things that I struggle with is trying to make people not feel guilty about sins of the past. And that's a tough one, right? Because we all have some sort of responsibility, even if we weren't here. But it's really more about what do we do next, right? So understanding the past in order to build the future. And so that's what I have to do.
Take your questions while standing in front of you.
Oh, yeah.
So like, we don't really know about this. But you can actually. really efforts, acknowledge,
Yeah, so super timely question. I just spent 7 days in Yosemite National Park with an undergraduate research group. And one of the interesting things they arranged for us to like meet with park staff and stuff like that. In the past couple of years, Yosemite has specifically hired 3 indigenous interpreters because they have a little, I don't know if anybody's ever been to Yosemite, but in the valley, there is this Miwok village. And it has a very tragic history because originally, obviously, they were exterminated from the land, but the Miwok weren't actually able to do some of their ceremonial practices there until about the 60s. They kicked them out again. But they're actively looking for reconciliation, so they hired these indigenous interpreters and basically gave them free rein to come up with the interpreter program. And the lady we talked to, like, she was real. So she was basically like, I'm gonna tell you about this stuff, it's gonna be depressing, I don't care, you need to know. I'm sorry that I'm ruining your vacation right now, but y'all need to know like what the truth is. And she did it in a very, I mean, she didn't do it in a super aggressive way, like I'm sort of making light of it. But I think that that is one example of the national parks taking a concerted effort to say, tell your story the way that you want to tell your story. We understand that it might be uncomfortable for some guests, but it's important for them to know. And they also recently, I think within the past 3 years, I think that was part of a whole package deal sort of thing. They reestablished relationships with the 7 affiliated tribes that lived in that area. And they built, they gave those tribes access to a part of the park where it is not advertised to the public. And they recreated that Miwok village over there. And so now they're able to do their ceremonial practices in a space that is not part of the public area of the park. And they're also inviting them into a council for like, I think, the ecological restoration of the park as well. So Yosemite was like, they were doing a really concerted effort, and I had not heard of that about any other national park then. So it was really interesting, yeah.
I also. I remember starting and I remember I didn't know a staff meeting, I remember the staff meeting about I feel like the battle there, she was really like just saying, and I'm doing that. And I also don't know the names. That's something I really think the way that they live is like very straight. I will, but it's a longer time. It was very strange about my thoughts, like they were like, oh yeah, it's things for long. Yeah, we just love it here.
Oh, it's the big, it's like a huge jutting out. Yeah, yeah, it's still not done yet.
Yeah, they were like just visitors.
Right.
And they're just taking a family weird or something. And they really didn't explain the actions.
So I do think that, so Deb Howland is the first indigenous person to run any national or federal agency. So she is the head of the Department of the Interior, but also the National Parks chief is also the first indigenous national parks chief. So I do believe that that's part and parcel of why we're seeing some of these changes. And I am hopeful that it'll get integrated into the national park system. Because again, I think that they are the jewel of our country, but we also have to acknowledge how we got there. For sure. Yes.
Do you know of any collaborative efforts like globally that are happening right now to combat some of these things? Because obviously in other parts of the world, some of these things are happening actively and in the present.
I don't know any like large encompassing global initiatives. I do know that there's a lot of grassroots efforts. There's also in some of the big NGOs, there's a recognition that the type of conservation they've been doing abroad has been more hurtful than beneficial. And I do think that they have, so for example, National Science Foundation, which is like the huge for grant making, they have also, I don't remember the name of the subdivision, but there's one where if you were to work with an indigenous group, you have to lay out a plan about how you are going to split responsibilities and acknowledge their sovereignty, which is something that wasn't part of grant making, 5-10 years ago. So I think it's slowly gathering steam, but I don't think that there's one big organization that's kind of out there doing this internationally. And I do think sometimes the local groups themselves have also said we've had enough, and so that has been enough to spark a conversation. I know that there's a couple of examples in West Africa where the locals have been essentially like, they have militarized the parks because of poaching, but poaching, they have to poach because their traditional sources of protein are in the park. And so they basically revolted in a couple of these instances and come to the government to say, like, this is not working. We need a new way of doing things. So sometimes it's more of an aggressive process than others. Yeah, sometimes it is more intense, more combative for sure. Yeah. Any other questions? Thank you.